Welcome to part 2 of my video mini-series, Does Soil Microbiology Work? Where I'm doing a test, exactly this. Does soil life have any effect at all on the plants that we grow? If you haven't already seen part 1, I would strongly suggest that you click on that now and have a look so that you fully understand what I did, how I did it, and the kind of results that we're aiming for here. I'll put a link to part 1 in the description below, and I'll see if I can link to it somewhere on the screen as well. And for those that saw part 1, just a quick reminder, what I did was set up some pots of soil with low levels of microbiology in and more natural levels of microbiology in. We've got some replicates, so we've got some measure of rigor to what we're doing. And in this video, we're going to have a look at the results in more detail. So without further ado, let's get stuck straight into it. Okay, so we're in the lab filming the final days. This is day 8 and we're going to harvest the trial pots. At the front here we have the sterile soil, not sterile, but the autoclaved soil that should be low in microbiology. And then in the pots at the back we have the kind of more natural soil. And you see I've removed them from their little trays. Interestingly, you can see that the roots on the more natural soil here are pretty solid and fairly proliferous, whereas on the autoclaved soil they are not protruding from the bottom of the pots. I'll do a better close-up. And here we go. In this hand is the kind of sterilised, and here is the natural, more natural soil with lots of biology. That's those two. You can see actually it's the pattern right across the board, which has kind of shown up a principle pretty well. So it's the same across all my replicates, stronger rooting in the natural soils. What I'm going to do now is harvest the cress and weigh the organic matter from this. So I'll set up a time lapse so you can see me do this. I imagine it's going to take a little bit of time, and I've got some results to record as I go. So we're now going to do the last of the samples, the last natural and the last kind of sterile pot, but I'm going to put this on actual record so I can talk you through what I see as I do it. And actually this is very interesting. We'll look at this quickly. You can see the roots in our sterilised soil here compared to the rooting system of our more natural soil here. Perhaps the camera doesn't pick it up, but there is actually quite a difference. So here we go, we're going to do the last sample now. So this is the last kind of sterile. I'll just trim the roots at soil level whilst trying not to spill any soil onto our tray. It isn't the most accurate way, but it will do for our purposes here. There's a little bit of soil I can see, so we'll put these on the scale now. We've already zeroed the scales. You can see what these come in at, and in case you can't quite see, there's a few stragglers here. That has measured at 1 grams. Okay, and And then we will do the last of the natural soils now. We'll zero that. Get our pot off of here, put it on the mat, and we'll do the same. So we're just trimming at soil level. And actually I have noticed that the more natural soil does seem to be quite a bit taller. There we go. Let's get the stragglers onto the tray as well. We missed the odd one, but to be honest with you, with the differences I'm seeing across these, I don't think it's going to make too much difference. And the final result of that one is interesting. So these are the samples from our harvest: natural, 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 sterilised, sterilised, sterilised. I have the pots left over here, which I'm going to put into this tin foil, and then we're going to get the guys in the laboratory to do a soil analysis on it tomorrow, and can look at the bacteria and fungi levels. So the final part of the test for the time being is to measure the nutrient content of these samples, which I'm going to do with this thing, a refractometer, which will measure the bricks. In essence, this is the amount of suspended solids in the sap of the cress that we're going to look at. So there we go, that is everything I did, including the harvesting of the cress itself and collection of the data. So let's get straight into looking at what the data showed us. You can see the 6 replicates, the 3 natural soil and the 3 reduced microbiology. They're the ones that we put into the autoclave. First thing to notice looking at the weight of the cress is the distinct difference between the natural grown and the reduced microbiologically grown. The averages actually were for the natural, that came in at 33 grams, and for the autoclaved soil the average was 26 grams. Now, kind of thinking back to when the plants themselves were growing, I did notice, and I kind of mentioned it a few times, that they did look like they were growing just that little bit quicker, the cress in the natural soil. Also looked a little bit greener, and I did make multiple reference to the root system as well, which were far more pronounced in the natural samples. Other kind of points to note is really the variation between the replicates. Now, these certainly are not kind of scientific replicates, but we did sort of a number of each pot just to have a little bit of rigor to what we're doing. And one of the things that I was kind of really struck by was the degree of variation between the largest and the smallest weights. Now, on the reduced microbiological, the autoclaved soil, the variation was 4.7 grams. So that was kind of the difference between the smallest or the lightest kind of cress sample and the heaviest. Whereas on the natural, that variation was actually down to only 2.8. So quite a big difference, to about 2 grams difference. So the natural soil appeared to deliver far more consistent samples as well. In retrospect, I did kind of think when harvesting the cress samples that perhaps I might have been better off actually extracting all of the plants from the potted soil and kind of washing all the soil off and including the roots as well in measurements, just to get a total organic matter measurement. But as it was, the harvesting was kind of done chopping the cress at soil level, and it wasn't the most accurate way to do it, admittedly, but to be honest with you, I didn't think it would make too much difference, and actually the weights themselves kind of reflect the observations really that I made at the time as well. So overall, when you consider these weights, actually the natural soil delivered just over 22% more growth than the low microbiological soil. So really quite a stark difference between the two. The bricks, on the other hand, looking at that data, again, it's very consistent across all the samples. It came in at 1.5% for all of the naturally grown cress samples, whereas the low microbiology sample was 2%, so slightly higher in fact, and that was the case across the entire board. Now, admittedly, when using a refractometer, the scale that you actually observe when you're looking through the eyepiece is pretty tiny, and so these are kind of estimates, but actually I think those numbers do reflect where I kind of saw the level every time I looked. So on that front, I guess it leaves a question: why might the bricks be lower in the more natural soil at this stage? So if you have any suggestions or thoughts about that, do stick them in the comments below. We'll kind of leave that as an open-ended question at the moment, because we've still got the soil analyses to do, so hopefully that will add a little bit more insight into why the difference in terms of the bricks values. So we're going to leave it there for this particular video. That was part 2. There is going to be a part 3 where I'm actually going to talk about the analyses that I had carried out on the soil samples. So we'll look at that in part 3. For now though, if you found this interesting or helpful, do give the video a like. If you find videos that give you tips, advice, and show you how to look after soil to get healthier, tastier food, then do consider subscribing to the channel, and we'll leave it there for today, and I look forward to seeing you in the next video.